right. Oh. Just ignore that. All right, so today is the third to last normal lecture, and then next Friday is going to be one of our normal review sessions. So keep that in mind. Put together study questions over the weekend. Make sure to send them to me next week so that we can include them on Friday's review. Today we are going to talk about human development, kind of in a more general sense, more about the basics of biology, of what's happening, including we're going to start with, I promised, a discussion about mitosis and meiosis. So we're going to go over that. I'm not going to go into every single step, all the individual parts, because this is something that you're expected to know from Bio 100, whatever form of that you took. But if you do have questions about mitosis or meiosis in more detail that I'm covering, I'm happy to go over that with you. Either you can send me an email, come to my office hours. I have slides from my human bio courses that I can send you. Just if you feel like you don't understand it, I'm happy to go into more detail personally. All right, so every person starts out as a single diploid cell. So they're going to have 46 chromosomes, and it's just a single cell. We've got a single nucleus here, cytoplasm, the various other organelles that are going to be within a functioning cell, things like mitochondria. Keep in mind that the mitochondria only come from the mother. Remember, the sperm, when it donates its material, it's only donating the genetic material. And so that's why mitochondrial DNA can be traced through the matrilineal line. It's because it's exclusively coming from the egg, coming from the mother. You're never going to get the mitochondrial DNA from the father. So the zygote is what we call the fertilized egg, so the fusion of the sperm and egg chromosomes. Once we've created this single diploid cell, we have to go from a single cell to something fully developed like we are, which is the hundreds of millions of cells. And that's just going to involve the process of dividing this initial cell over and over and over and over and over and over and over again until we have a fully functioning developed human. So in order to talk about that, we have to talk about the cell division process. Cells are going to normally divide by the process of mitosis. Remember, this is for all of your somatic cells, anything that is not a gamete. It starts from a diploid parent, so 2N, meaning it has two full sets of chromosomes, a total of 46 chromosomes. The first part of this process involves duplicating the DNA, and then we're going to divide that cell into two new ones. You have to duplicate the DNA because otherwise your two daughter cells are going to have half the DNA of the parent. So you need to duplicate it first so that when you separate it, you end up with two fully functional cells that have a full set of chromosomes, or full two sets, excuse me. So the result of a single process of mitosis is two fully diploid daughter cells. These are going to be identical to the parent. So this is a key difference between mitosis and meiosis. Mitosis always results in daughter cells that are identical to the one you started with. Like I said, we have to start by duplicating the DNA. That means we have an extra copy of every chromosome. So during this phase, during this moment in mitosis, you're going to actually have more like 4N. You're going to have duplicated everything. And these copies we call sister chromatids. There's nothing particularly special about the change of material or structure here. It's just a duplicated pair of chromosomes. So we start with a single chromosome, they go through duplication, and we end up with two copies. We call these together the sister chromatids. They typically form a bond in the center called a centromere, which gives them this very X-like shape. Chromosomes aren't naturally on their own X-shaped. So when you see those karyograms that I've shown you before, where you see the lineup of all the chromosomes, and they look kind of like this, those are taken at the point where it's already duplicated and it's in preparation for the separation. A single chromosome should look more like a rod. So once we then go through this division process, we're going to separate the sister chromatids. You get 2 individual chromosomes that can then be sent into the 2 daughter cells. All right, mitosis. Typically, mitosis is broken down into 4 stages. Sometimes people will include a 5th stage. I typically include that 5th stage as part of the 4th one, but you can kind of subdivide it however you would like to. And if I were to ask this on an exam, for example, in a short answer question, if I asked for the 4 stages and you want to give me 5, that's fine, as long as you're specifically separating that last step into the 2 stages that I'm going to talk about. So these are the 4 names: prophase, metaphase, anaphase, telophase, and then cytokinesis is the last one that kind of falls into telekinesis, my goodness, telophase. This is the proper order, so you can learn them in this order as well. So our DNA has already duplicated. I'm kind of skipping over that step. Again, I'm simplifying the process here. So we're going to start with the actual process of beginning to divide for mitosis, and that is prophase. All of those sister chromatids are condensing. They're contracting into a single location within the nucleus. Once they have all contracted, the nucleus, the nuclear envelope, dissolves. So we're removing that barrier that normally keeps the chromosomes, all of that DNA, safe from external material. We're removing that barrier. We also form these structures we call centrosomes. These are just little kind of poles for the cell, and they're going to generate mitotic spindles, which are just tubes. They're essentially suspension bridge, like that kind of stretchy piece that's going to hold the parts together. Those are going to form and create this bridge between your 2 poles, between the 2 centrosomes. So that's prophase. We're just putting everything in place so that when we do start to divide, everything's where it needs to be. Metaphase comes next. Those centrosomes fully separate and migrate to each end of the cell. So this is why I talk about them as being the poles of the cell. You're going to get one on each end. They stretch out those mitotic spindles, and they're going to grab onto that centromere, that center point of those sister chromatids. That's kind of the hardened point of the chromatids. And they're going to pull just enough that all of the chromosomes, all of these sister chromatids, are going to be lined up down what is essentially the equator of the cell. So they line up down the middle stretch. And now we can begin the process of actual division. So now, during anaphase, those mitotic spindles pull. It's an abrupt pull, and that's going to separate the sister chromatids at the centromere so that one of each copy is going to end up towards the north pole and towards the south pole of your cell. Once you have separated the sister chromatids, these are now chromosomes again. It's just a change in nomenclature. And those are going to be pulled towards each end so that when we fully subdivide right through the middle, each resulting cell is going to have one full set of all the chromosomes it needs. Okay? And then finally, telophase, and like I said, I typically include cytokinesis here because they're technically happening simultaneously, so it's, for me, I don't really see a point in separating them. At this point, you have pulled all of your chromosomes towards one end or the other. They start condensing again, they start forming a center, and that allows us to reform a nuclear envelope. So we've recreated that barrier now. We're going to protect all that chromosomal material. The centromeres start to decay as well as the mitotic spindles. Those are going to be recycled because the cell no longer needs them for the current moment. And these first parts, this is going to be technically the telophase. That's going to be the reforming of the nuclear envelope, the separating of the chromosomes, and the redissolving of the mitotic spindles. And then while that process is happening, cytokinesis is happening, and that's where the cytoplasm starts to pinch, starts to squeeze right down the center, and eventually that squeezing will reach each other, and it just pops them into 2 separate cells. So cytokinesis is just the division of the cytoplasm, separating them into two individual daughter cells. Okay. Just a little bit more about cytokinesis here. So in animal cells, this happens differently in other organisms, but in animal cells, cytokinesis happens through this process we call cleavage. The way I like to visualize it is if you take like a water balloon and you press down on one side with your finger, that's going to start forming kind of a cleave, a divide between them. And if you can then picture that cleave going all the way through until it pops out the other side and creates two mini water balloons, that's what's happening during cytokinesis. So it creates this cleavage furrow, which will then push through all the way to the other side. So the result of this process is you will have two fully diploid daughter cells, and again, they are identical to the parent cell that you started with. All right, so that's the process that all of your somatic cells are going to go through, and they're doing it all the time, replacing your cells. That is the basic set of steps. We are also going to run through meiosis. I've already talked about it in general terms in a previous lecture, but just to run through the steps again so that you know them, it is very similar to mitosis because it evolved from mitosis. Mitosis was the original form of cell division, and then in sexually reproducing organisms, we evolved this extra step. So the phases are all essentially the same with a couple of key differences. Meiosis is only going to be used to make haploid gametes, so again, only for sex cells. And one of the key differences is that it's going to involve 2 rounds of division so that you end up with 4 cells rather than 2 as your end result. Just like mitosis, we have to start the process with duplicating the chromosomes, so we're going to end up with twice as much DNA. And then, like I said, there's going to be 2 phases of division, and they're named quite simply meiosis 1, that's the first division, and then meiosis 2 is the second division. So at the end, the products, we're going to end up with 4 haploid cells. So you're going to get twice as many cells, but each are going to have half as much DNA. And the other key thing is that these haploid daughter cells are going to be genetically distinct from the originating cell. All right, so to run through the phases, they're the same phases as we saw in mitosis: prophase, metaphase, anaphase, telophase, and cytokinesis. We do give them a 1 if it's in meiosis 1, and then they will have a 2 if it's in meiosis 2, just depends on what phase. And again, they operate in the same way as mitosis, so I'm not going to talk about what's happening in prophase 1, or anaphase 1, all of that, because I just went through it. It's the same process. Instead, I'm going to focus about what's the key difference, and the big one happens almost immediately. So we've duplicated our DNA and we've started meiosis 1. We are in prophase 1, the very first step. This is where crossing over happens. So I've talked about crossing over a little bit already. This is where you have your homologous chromosomes. They meet up next to each other. They break off pieces of their genetic material, and then they swap them. So they're exchanging that genetic material. So this happens immediately during this process of meiosis. So here's just a zoom in of that so you can see we have red and we have blue sister chromatids. They meet up. They're going to exchange that genetic material. And you can see as we go through the various phases, once we start separating them, some of them have blue with a little bit of red, some of them have red with a little bit of blue, and varying amounts of each. Okay. So that's the big difference during meiosis 1, is that we have that crossing over phase. Meiosis 2, like I said, exact same set of phases, and then we just add 2 to all of them. And so what we're going to start with is, rather than starting with a single cell we're starting halfway through this division process. We've already divided once, so we're starting with 2 cells. Each of those are going to go through a division phase so that by the time we reach the end, we have 4 separate haploid daughters. So the key difference here is that meiosis II starts with 2, ends with 4. Okay, so here's a nice summary slide of meiosis. I'm not going to go through all of the individual details here. Again, this is hopefully mostly review, but it's here for you if you need to. Starting with meiosis 1 with prophase 1, we get our crossing over happening, and then you just go through the normal phases of division, and you just do it twice. All right. Meiosis ends the same way as mitosis does. So you're going to have that cytokinesis through the process of cleavage. That's going to separate the cytoplasm. And again, we're going to end with 4 daughter cells. They are all haploid. They all have a half set of chromosomes. And then each daughter cell is going to be genetically distinct. This is the key difference, and this is going to promote genetic diversity within a population. So this is just a context thing. So every egg that is undergoing this process, all of the, well, not egg, but the oocytes that are going to become eggs that are undergoing this process, every egg that they produce is going to be genetically distinct. Same thing goes for males. I mentioned they're producing a couple 100 million sperm per day. Every single one of those sperm cells is genetically distinct from the other. That's why even if your parents have 20, 30 kids, every single child is different unless they are identical twins in some way. So you get this genetic variation because of this process. All right, so going back to crossing over, just to put this in context here. So after the duplication phase, during this prophase 1 of mitosis is where we have the crossing over happen. So the sister chromatids line up and they exchange a little bit of genetic material. And then synapsis is just the term for where we repair those breaks. So we've broken it, we've exchanged material. You have to repair that connection so that it's a single continuous strand of DNA again. We call that process synapsis, and you just rejoin those exchanged pieces of genetic material. So this is important. I've already stressed this because it provides genetic diversity. Because we are sexually reproducing organisms, we are capable of this process. If you are a cloning organism doing asexual reproduction, all of the produced offspring are going to be genetically identical to the parent, which becomes detrimental as far as evolution goes. You don't provide variation in your population, and if there's changes in your environment, you have less ability to adapt to that environment. There's less variance for natural selection to act upon. This is also important, I mentioned, because crossing over is capable of unlinking genes. So remember, genes normally follow the law of separate inheritance, but if they are on the same chromosome, and especially very close to each other on the same chromosome, they can become linked, and they will be inherited together. Crossing over can break that linkage, so we can separate that genetic link. And then creating, of course, new combinations of alleles so that every produced offspring is genetically distinct. Ooh. Okay. I have a meiosis summary slide. Again, I'm not going to go through this in detail. I do want you to know the 4 or 5 phases of mitosis and meiosis, but what I really want you to focus on are what are the differences between mitosis and meiosis. So how are they used differently? What are the main differences in the process? So this slide summarizes that for you as just a review thing while you're studying. All right, so we're going to jump back to where I started in this lecture. We start as a single diploid cell, the zygote, the fertilized egg, the fusion of genetic material from the sperm and the egg. So this zygote is now going to undergo mitosis because we don't need to be creating gametes from this cell. We just need to be making more somatic cells. And it's going to do so through this process of repeated mitosis, repeated cleavage, separating that cell into more and more cells. The key here is that our zygote isn't increasing in size for quite some time. So all of this division of subdividing our original cell into 2, 4, 8, 16, 30 some cells, this is all happening without any growth of our zygote. It is staying the same size. This is all going to be happening while still within the fallopian tube. So I mentioned that our fertilized egg happens very close to the ovary. It's at the beginning of the fallopian tube. And once that egg is fertilized, it's going to be released and can start to migrate through the fallopian tube, but it's a slow process. The zygote does not have any type of flagella to propel itself, unlike the sperm. But there is a very slow but natural drift of the fluid that exists within the fallopian tube, and it creates a current. And so the zygote will be gently pushed along this structure. While it's being passed through the fallopian tube, we see this subdivision. We see the cleavage happening. So we're creating increased numbers of cells, but we are remaining at the same size. This whole traveling process takes approximately 7 days. So from fertilization, it's about a week before it's going to reach the end of the fallopian tube and can then be implanted within the uterus. By the time this dividing zygote has reached the uterus, we can call it an embryo. And the embryo is a hollow ball of cells. So we've subdivided a whole bunch. We've created a numerous cells, but they haven't increased in size. We call this new structure the blastocyst. So here we have our zygote. It's subdividing multiple times. By the time it reaches the uterus, it's subdivided. We have many, many cells, but it's hollow on the inside. It's also divided the types of cells now. So we've created a distinct portion on the inside of the blastocyst called the cell mass. Those cells are going to be the ones that eventually do grow into the new organism. The rest of the cells, the ones surrounding it, creating our shell, those are going to be providing nutrients for the cell while we are developing the placenta. So the blastocyst reaches the uterus. It's going to implant itself along the wall of the uterus. At this point, we are in the secretory phase of the uterine cycle. So the endometrium is as thick as it is going to be. It's going to be full of those small arteries. So we have blood supply that's going to be providing nutrients. And it can attach to that so it can start taking advantage of those provided nutrients. Again, this is going to be several days to a little over a week for this implantation process to happen. Once implantation actually occurs, once that blastocyst has made contact and is attached to the endometrium, that is the beginning of gestation, which is the proper term for pregnancy. Now, our embryo, our little blastocyst, is going to start infiltrating that endometrium. It's going to start sending those surrounding cells into the surrounding tissue. So it's increasing, it's continuing to divide, it's creating more and more of those surrounding shell cells, and those are going to move into the endometrium. The reason for this is that they need to integrate the arteries that were created within the endometrium into their structure. So they need to have access to those arteries in order to get the blood supply, in order to get the oxygen and the nutrients that they need to supply them. So you can see that happening in this picture. We have our blastocyst making contact. It's starting to migrate these surrounding cells into the endometrium. They actually physically envelop those arteries so that they can integrate them into their own system. This process is what will eventually evolve into the placenta. So this combination of the blastocyst cells surrounding the arterioles of the endometrium. It is not a placenta yet, it is still just a combination of tissue. We also start to see a specialization of the materials within our blastocyst. So the embryo is going to differentiate into that cell mass that is actually going to become the fetus. Those are the cells that will eventually become the organism. But it also creates this larger structure we call the yolk sac. Now, this is not to be confused with the yolk of a chicken egg. They are fundamentally different structures. The yolk of a chicken egg is the nutrition. It's the material that is being supplied to that developing organism as it grows. It's no longer attached to the mother, so it's not getting any of its nutrition through that method. So it's entirely from that yolk. In humans, the yolk sac is more analogous to like a primitive organ. So it's going to be the attachment to the mother, and that's how it's going to take in the oxygen that it's getting from those arterioles, how it takes in the nutrients and provides that to the developing fetus. So it's a primitive organ that will eventually be taken over by the placenta. So, not a chicken egg. So we've created this structure. We have blended both mother and fetal tissue. We're starting to access that oxygen and nutrition. And we've started to specialize and divide the cells within the developing embryo. We create 3 different layers in this process. So we have this, one side is the yolk sac, the other is the amniotic cavity that will eventually become the amniotic fluid. And in between them, we sandwich 3 layers of cells. We've seen them before, but just to reiterate, these are called germ layers. The outermost is the ectoderm, the middle is the mesoderm, and the inner is the endoderm. Now I say outer and inner because that's what they're going to become. At this stage, they're just linear. They're just sitting one on top of each other. The ectoderm is closest to the amniotic sac, and the endoderm is closest to the yolk sac. So if you look at this more cartoonized version, here's your 2 sacs, and then the linear division of these 3 cell types, or 3 cell layers. It is these germ layers that are going to become the new organism. So we have to change them slightly. We can't just stay linear. We have to create a 3D organism, and they're going to do so by folding in on themselves. So you get this bump up. The yolk sac pushes up into that set of tissue and folds them into a tube. This is going to create that fundamental organization, so it creates an outside and an inside. Okay. So a couple more pictures just showing you a very similar thing. This whole process of creating the tube that will become the organism is called gastrulation, so the folding of these germ layers. The yolk sac, like I said, is what's going to be doing the actual folding, so it pushes up into those germ layers and forces the ectoderm and the mesoderm to fold around it. So again, another visual. Water balloon is a good one. You take your water balloon and you start pushing into it with your finger. It physically has to fold around that structure. This will continue to happen. We'll continue to get this enveloping until finally the ectoderm and the mesoderm completely surround the ectoderm on the endoderm on the inside. Whew. I have another visual. I always think about it like a little sea anemone. If you've ever played with them at an aquarium, if you poke your finger in the center, they wrap themselves around you. It's a very similar analogy because they actually develop in the exact same way we do at this stage, and then we eventually split in very different directions. So the goal of gastrulation is to create this layered tube. And again, just showing you, we have this pushing up of the inside layer, creating this fold, which can then be fully enveloped to create a tube, which will become our organism. There, too many animations. This poking in that we see of our inner layer is actually what will become the anus. So this is the first opening that is created for humans. This is not always true. There are different groups of organisms that make their mouth first. We are part of Deuterostomia. That's just the group of organisms that make anus as the first biological opening. Eventually, that pushing in is going to push all the way through, and that will create the mouth on the other end. We are biologically tubes of meat. We have a hole that starts at the front and it goes all the way to the other end. And this is how it happens. All right, so now we're going to talk about what happens to each of those germ layers, because they are each going to specialize to create a different layer of our organism. So we end up, after this gastrulation, with our cylindrical tube. We have our tube running through our center, and we need to specialize each of these 3 layers. We will start with the endoderm. So this is the inside. This is the part that has created that tube, which might indicate to you what types of tissues it's going to turn into. It's the material that is the deepest within our body and the material that forms the tube that goes from the front to the back. So it's going to be our gut tube, our entire digestive system, starting from the mouth all the way to the anus. It's also going to develop into the urinary system as well as the respiratory system. The respiratory system should also make sense because it's attached to that center tube. So the endoderm becomes your innermost tissue, the stuff that's at the core of your organism. Here's another visual showing you that tube. So we have the very beginning, our opening, we have the very end at the colon and the rectum, and it just creates this big tube running through the center of our body. That is what our endoderm becomes. It's funny to think of yourself that way, but you are a tube. All right, mesoderm. This is the middle layer, and fairly logically, it becomes the stuff in the middle. So it's going to be the stuff surrounding what the endoderm became, but before it becomes the ectoderm material. So this is going to be your skeletal system. This is going to be kidneys. Those don't quite match up with the urinary system. They're built separately. All of your vasculature and all of your musculature. Does that make sense? That's kind of the stuff that's in the middle, going from the core outwards. Oh, here's a summary slide for you. So here we have our endoderm, what it becomes. Here's our mesoderm, all of the things that it's going to become. And then we can move on to the ectoderm, which is the outermost layer. So we've created that tube. This is the outside structure. So it's going to be the outside plus something extra. So the ectoderm becomes your skin. This is going to be your outermost layer. It's going to include all of the structures that exist within your skin, so all of your sweat glands and all of that. But it also becomes your nervous system. So the ectoderm is responsible for creating the brain, the spinal cord, and all of the nerves that run through your body. So that's the only part that doesn't really fit the normal pattern. Okay. So here's that summary slide again. Here's our ectoderm and the pieces that it is going to become. All right, so that early stage of creating the tubal organism is pretty much the same for just about every single animal on Earth's surface. They all have a very similar development in those early stages, which is why once you've created a fetus, that tubal organism, if you look at a number of different organisms, they actually look very similar to each other in these earliest stages of development. And the same can be said then that a human fetus at these earliest stages of development doesn't really look like a human. Doesn't really look like any particular organism. They look more like a convoluted tube. After this phase, we can start to develop towards more human-like characteristics, developing the human organ systems in the particular organization that we use compared to other organisms. All right, so this is what I was getting at. During those very early stages of development, all vertebrates look very, very similar. And that's because we all share an evolutionary origin. We are humans, which evolved from tetrapods, which evolved from fish. So we were all fish initially. That's how various forms of life all began. Everything was a fish-like organism. And so our earliest stages of embryology represent that. This is a lizard, this is a tortoise, this is a pig, this is a human, all at the same stage of development. And they're nearly indistinguishable. It's only as that fetus continues to develop that you start to get differentiation into what is clearly a different organism. Okay. So I'm going to talk a little bit about fetal blood supply. I've talked about this before when I talked about cardiovascular system at the very beginning of the semester, but I think it's interesting and it's part of fetal development, so throw it in here. Fetuses get all of their oxygen from their mother. They are not exposed to the atmosphere, so those lungs are not functioning as lungs during gestation. That means there's no real reason to send blood into the lungs of a developing fetus. So you get the placenta that's providing all of the oxygen and nutrients from the mother. It passes all of that through the umbilical cord, which is going to pass first through the liver. Filtering can happen if necessary, although that's not typically needed. And it exits the liver. If you remember what happens in our discussion of where the blood passes once it's been passed through the liver, it joins up with the inferior vena cava. Now, the inferior vena cava, is that oxygenated or deoxygenated blood? It's deoxygenated, right? And that's the truth for a fetus as well, because the oxygen has been used up as it's passed through the lower extremity. So we have deoxygenated blood, which is then being mixed with oxygenated blood from the mother. So the blood of a fetus isn't fully oxygenated. It's never going to be at 100% of a normal adult human. That partially oxygenated blood is sent up to the heart, follows this normal pathway up towards the right atrium, and then it doesn't need to go into the lungs, so it doesn't need to follow the normal pathway through the heart. I got a little ahead of myself here. Here's the part about the liver and the inferior vena cava. So it gets sent into the right atrium. That's our normal entry point. But then rather than going into the right ventricle to the lungs into the left atrium, there's actually a hole that exists in a fetus between the two atria. We call this the foramen ovale. And the blood can pass directly from the right atrium to the left atrium. At that point, it will follow a more normal pathway, so it'll go from the left atrium into the left ventricle, and then out through the aorta, sending oxygenated blood to the rest of the body. So during development, during gestation, a fetus's heart actually functions more like a 3-chambered heart than a 4-chambered heart like we see in a full-grown adult. That hole, the foramen ovale, closes as soon as that baby is born. That first breath of air drastically changes the pressure inside the thoracic cavity, and that physical pressure change causes a little flap of skin that exists on the side of the foramen ovale to slam shut. And that will then fuse and form the walls separating the atria. I talked about again during the cardiovascular discussion that the flap closing isn't always 100% perfect, and so individuals that have gestational holes in their heart, it's because that flap didn't close completely. Sometimes that will close on its own, sometimes surgical intervention is needed. So here is a zoom in of that same process. Again, we have the blood coming from the mother, oxygenated, passes through the liver of the fetus. It then gets entered into the right atrium, skips over the right ventricle, straight into the left atrium, left ventricle, out through the aorta. All right. So birth is, of course, the final stage of gestation. This is where we are going to sever the direct reliance on the mother for all of its nutrition, oxygen, all of that. During gestation, all of that's coming through the placenta and being sent into the developing fetus. after it's born, we sever that direct connection. Now, we as humans are not precocious. We cannot just get up and run the moment we are born. We do still rely heavily on our parents for survival. And part of that is milk production from the mother. A female is capable of producing milk from her mammary glands. We'll talk a lot more about this on Monday when we talk about pregnancy, but that is going to continue to supply nourishment to the developing child for the first stage of life. It also assists in adding immune system defense. Again, we'll go into this more on Monday. Development does not stop once birth occurs. Obviously, we don't stay babies, we grow up. So humans continue to grow and to develop and to change for the first 16 to 20 years of life, depending on the individual. Most people reach their final stages of growth somewhere in their late teens, early 20s. We don't just change in size, however, we change in shape. Humans are part of what we call allometric growth trajectories. So there's isometric growth, which is just where you change in size, but the shape changes the same, or the shape stays the same, does not change. Allometric growth is a growth in size as well as a change in shape through development, which it makes sense. You may not think about it, but a human baby looks very different from a human adult. Our proportions, our relative sizes of the different regions of our body change as we develop. So here is a cartoon image. This is a human infant, and proportionally the head is about the same size as the thorax, is about the same size as the kind of hip region and the lower leg. They're fairly evenly divided. If you took a human infant and made it an adult size, they would not look like an adult human. Their proportions are very different. Instead, we change those proportions. Our head becomes proportionately smaller to our torso, our limbs become proportionately longer. There's a change in size. So we're doing this allometric growth. Or excuse me, change in shape. This is allowed because we have parts of our skeleton that don't immediately fuse upon development. We have these epiphyseal plates, which are cartilaginous layers within your bones that continue to allow these bones to lengthen and potentially change in their particular shape as an individual gets larger. So these are found in mostly long bones. Remember way back when we learned about bones, long bones include most of your limb bones as well as your fingers. Your fingers count as long bones. And so if you look at an X-ray of a child, someone who's not reached their final stages of development, what you will see are these lines near the ends of those long bones. Or in the cases of the digits, the fingers, there are these little pads towards the end. Those are going to allow for the growth of those bones. Once an individual has reached their final size, those epiphyseal plates fuse, so they go away. It is actually something that can be used to age a skeleton, whether or not that epiphyseal plate is completely present, in which case those two pieces of bone can be completely removed from each other. If it's partially fused, that happens at a particular range of age, or if it's completely fused. So you can use that to age a skeleton. And then the final stage of human development is puberty, which of course I've already talked in length about, but it represents the final stage of human development, which is the ability to then reproduce and produce the next generation, which evolutionarily speaking, that is the pinnacle of life. That is what is supposed to happen. We're supposed to reproduce and make more humans. But we see accelerated growth, again, the exaggeration of secondary sexual characteristics, and that onset of fertility. So that is human development. Your basic topics or key topics, basics of mitosis and meiosis, what are the stages, the basics of what are happening at each of those stages, and make sure that you understand the differences between them. The stages of embryonic development, the germ layers, what do each of those become, and then fetal circulation, since I covered that at the end. And then Monday, like I said, we're going to cover pregnancy. Wednesday is more of a fun lecture. I'm going to talk about functional morphology and kind of the unique things that humans have evolved just because I can. And then Friday will be a review.